and I'll turn it over to Fred.
Sounds great. Thank you for the introduction, Natalie. I got it. That Zoom is recording. Okay. So welcome today to the AnVIL demo. We're focusing on combining data across multiple workspaces. And what I'm showing you right here is a AnVIL workspace that tries to collect the information for this demo. We're still working on making this publicly accessible, so stay tuned for links and updates. But know that, as Natalie said, all this information will be available after the fact, whether you're here or you're joining us afterwards. So I'm going to start by just giving a high-level overview, which is from our series AnVIL in 2 minutes. And in this workspace, you can see right here, there is actually a link to the slides. And I'm not going to go into full screen just because I sort of lose some of these Zoom controls. So hopefully this size is large enough for people. But the fundamental question we're trying to address here in this demo is, how do you analyze data across multiple workspaces? AnVIL hosts many high-value datasets, 1,000 genomes, GTEx, CCDG, HPRC, Human Pangenome Reference Consortium, which we'll be highlighting today. And some of you may even have, or many of you may even have your own datasets that you'd like to analyze jointly with these. So the question is, how do you do this in the most efficient and AnVIL-centric way? So there's two possible ways of doing this. One, which is maybe more traditional, is that you make a copy of all the data that you care about, and then you do the analysis on your cluster, on your laptop, what have you. The challenge of this is that it creates multiple copies of the original data and is not necessarily the most efficient way of doing this. Where AnVIL steps in is trying to invert this model, where rather than bringing the data to you, you bring your tools and your analysis to the data. So how can you point your tools to the original data? Cloud computing is one great solution to this. So to give an example that we like to talk about as a real world example is the 1,000 Genomes project, where you have many human genomes coming across different superpopulations representing people across the globe. And so as for example, maybe what you have is samples from the Americas, from East Asia, and you want to first ask the question, is this data good enough for my study? You may want to run a workflow where you run a quality control analysis, and that's going to be the example that we're going to show to you today. So the way that you would do this is take advantage of something called data tables. I'll be showing this in a little bit, but the idea is that you can think of this as a spreadsheet which keeps track of the location of the files you're interested in, whether it's your files, whether it's files from one cohort, files from another consortia. You want to organize it in a table. And this table then points your subsequent workflows to the original locations. Again, shown here is one superpopulation, and shown here is another superpopulation. But you can imagine these are different consortia. 1,000 genomes, HPRC is what we'll be talking about today. Now, how do you create this table? You can use any spreadsheet software you use, but we're going to highlight today a package that the bioconductor community has made, aptly called the AnVIL package. With a couple lines of code, we'll show you how you can quickly pull information from various AnVIL workspaces, which organize different consortia datasets, and create a new data table so that you can then use and run your workflows. Once that table is created, you can select a workflow of your choice. In this case, we'll be, as I mentioned, showing you the quality control analysis, and we'll show you some of the outputs as well. So I'm going to stop this introduction here, and what I'm going to do is I'm going to go into our slides for this demo, which are going to introduce us to the exercises. These are built as hands-on exercises, but you'll be watching in this case. Again, we'll provide information for how you can do these on your own after the fact. I'm going to prepare my workspace and cloud environment so you can get a feel for the steps that you'd have to do. And while we're waiting for that compute to provision, discuss a couple more, again, just a deeper dive into the AnVIL data flow and highlight some notable doc store workflows. So again, for those of you joining just now, the goal of this demo is to show you how do you compute at scale, especially combining data across different workspaces, different locations, whether it be bringing your own data, data from consortia like GTEx or CCDG. The fundamental goal is you want to organize it in a data table And then your workflow will use this data table to point to all the compute resources on the cloud, what files it's going to manage the copying and both back and forth between the compute and the ultimate storage locations. So the two things I need to do is, one, I need to make a copy of this workspace. I need to clone it. And two, I need to launch a Jupyter notebook environment so that I can show you how to create these data tables. For time's sake, I've already made a clone. I'm not showing you that here, but again, our instructional material will show you how to do that clone. What I'm going to show you is how do I start up a cloud environment. It's just one of those initial steps that people ask for, so it's worth us showing. I can either access it here in the environment configuration, or I can go into the actual analysis tab. I'm just going to click on it here right now, this icon. It gives me an option to start up Jupyter, RStudio, or Galaxy. I'm going to start Jupyter in this case. And you can see that this is going to cost about 5 cents per hour, pretty inexpensive for the power that you get through this. I'm going to use the default application configuration, which gives latest versions of R, Python, and GATK in this case. The standard CPU and memory is usable here, but you can go up to more than half a terabyte of memory with the current offerings. Same for disk. The 50 gigabyte is reasonable, but you can get terabytes as you need. So I'm going to click Create, and this is going to start provisioning that cloud environment. While this is taking place, which typically takes about 3 to 5 minutes, we're going to discuss a couple more concepts from this slide deck that you'll have access to. So again, what is a more efficient way of working with data? Rather than making a copy of the data that you care about, how can we just point to the data that we care about? AnVIL offers Terra data tables. Here it's listed as workspace data tables. And the idea is that you want to have URIs, universal resource indicators, pointing to where the data lives. If it lives in Google Cloud, you'll see that oftentimes it has, I think, this GS prefix. And so again, if you think of a data table just as a spreadsheet, you can imagine that each row would specify an individual file. So for instance, the first row could be pointing to a file that's located in this storage bucket with an appropriate file name. Here's an example of what you'll see when we start poking live through the workspace. Here's a sample table where we have 3 samples. We have the subject IDs relative to the consortia, and then this here is just the actual file name, but in behind the scenes, it's actually that GS, that URI to the Illumina sequencing data stored in a CRAM format. So this data table can point to files that are located in your own storage location, which would be bringing your own data. It could be coming from another consortia, this represented by this bucket, or it can be using the new GA4GH DIRS standard, which stands for Data Repository Service. It's an initiative that the NCPI group has been creating. It allows you to interoperate between AnVIL datasets, Biodata Catalyst datasets, NCI datasets, and more. So it's a great way to show the interoperability as you move between different consortia and resources. Once that data table is created, you can now start running your workflows. These workflows will spin up different compute environments, and that compute environment will simply get the location of the file, and then AnVIL will take care of all the copying into the temporary location so you can do your compute and then get rid of that so you don't have redundant copies. So again, one of the great things that AnVIL does is it orchestrates all of your cloud environments, orchestrates the copying, localizing, delocalizing of your files as necessary. And again, it can happen either from bring your own data or for any different consortium. So these 3 slides, you'll see that are familiar. I'm just recapping again in your mind. What are the steps? What's the process that you would typically do to combine data across different workspaces? The first is you create a data table. I'm going to show you how to use this using the bioconductor AnVIL package. And then you launch your workflow, in this case, quality control analysis workflow. So one final comment about workflows before we get started and I show you how it is live on the AnVIL platform. And it's what kind of workflows are available, because I'm talking about this QC analysis pipeline that the CCDG consortium has made. It's a great way to find out just what kind of coverage do you have, how high quality is the data, is there contamination? But once you've actually found that these datasets are useful, what else is available? And I'd like to just mention that there's an increasing supply of production quality workflows. The first notable case is GATK. So for those of you who are familiar with variant calling in genomes, GATK is one of the gold standards, and they have the best practice pipelines that recommend you use the not only whole genome and exome, but also increasingly additional research areas such as mitochondrial SNPs, calling germline variants from RNA sequencing data, copy number variations, as well as somatic mutations. So all of these great pipelines that you can immediately take advantage of once you start using the AnVIL platform. Another extension of this is the WDL analysis research pipelines, also known as WARP pipelines. And beyond just the germline variants, you also have access to genotyping arrays, single cell transcriptomics and epigenomics, and more. As consortia request more of these pipelines, and in collaboration, these are developed, they become immediately available to anyone who's using this platform. And then finally, just as an example of a group that's independent of AnVIL and the Terra communities, the ENCODE community has published their uniform analysis pipelines, which all can run also on AnVIL. And these extend beyond what I mentioned into modalities like ChIP-seq, ATAC-seq, RNA-seq. Again, they're very interested in the function of different elements in the genome. And in addition, chromosome structures such as Hi-C, and also whole genome bisulfite sequencing. So again, many great pipelines that you can start exploring, and DocStore is going to be your place to go. If we have time at the end and people are interested, I can also walk people through how DocStore is, but know that that's where you can find many of these. So what I'm going to do right now is I'm going to go over a couple of exercises that we have in this demo, which is exploring a workspace, actually creating the table, the dataset catalog, how you can find datasets of interest, and how you can find workflows of interest in DocStore. So at this point, I'm going to switch to this original workspace here to just orient people to where things are. So again, this is a Terra workspace. And in addition to this, sorry, is there a question there? Okay, if there's a question, I think the Zoom raise hand, that one's a little bit easier for me to see. So feel free to raise your hand if you have a question. Again, we'll have half an hour at the end to field many questions, and I can show more aspects of this as well. Okay, so a Terra workspace, often this dashboard here, is a great place to look because it'll have information. As I already demonstrated, we have links to slides and a video version of this so you can understand what's the goal of this, as well as where can you get more information, where can you ask questions, notably the AnVIL support form. I'm going to click on the data tab because this tells us what kind of data is being used in this workspace. If you look in the sample table, this table shows that we have 4 rows, 4 samples that we'd like to analyze here. And immediately you can see that on the project column, 2 of them are coming from the Human Pangenome Reference Consortium, HPRC, and 2 of them are coming from 1,000 Genomes. This cram column here lists the locations of each of those files, and notably, they're in different locations, right? So all of this here is organizing the analysis that we'd like to run. Now, what analysis are we running? The Workflows tab here shows the QC analysis pipeline. If you click on this, you can see that there is a location to get more information about this. We're going to click on this, which opens up DocStore. And here you can see more information that the authors are providing. They're showing you that this QC analysis pipeline will take your input file and it'll run several tests to make sure that it's sufficient quality. And then depending upon the thresholds that you set, is there sufficient quality? Is the contamination low enough? Is the coverage high enough? whether it passes or fails the relevant QC metrics. And different consortia have different thresholds, and you yourself could have different thresholds here. Fundamentally, all of these are being calculated for you, whether it's 1 sample, 10 samples, or 100 samples. So I'm going to go back now to, I'm going to close this and go back to the workspace and take a look at what are some of those metrics that are being calculated. And you can see again this in the data table in the sample QC results. So here there's more than 3 columns because now we have the sample and the CRAM, but now we have all of the QC outputs. I'm going to click on the settings because it's a little bit easier to see what's available across all the columns. But you can see that, as I mentioned, one of the quick things you might want to know is what's the coverage. So you have the mean coverage across the genome. You can also ask how uniform is that coverage? What percentage of the genome has 30x coverage versus 10x coverage? What's the insert size? How many look like chimeric reads, and et cetera. All of the things that the CCDG consortium has made is now readily available for you. This data table here organizes all the output, and I can show you that, for example, the evaluated metrics, these are the thresholds. You can click on this, and it'll give you a preview of the file. Since it's a TSV, you can easily see it. And you see that it passes all of the criteria that we've set, that they have set. In addition, if there's PDFs, you can see here, for example, the GC bias, you can click on it and you can see the actual plots that were made by this workflow. So again, it's a great way for people to quickly evaluate each of these files, each of these samples to see, is it good enough for your analysis? And then finally, one question that you might be asking is, okay, well, how long did it take? How much did it cost for each of these samples? Job history allows you to look at this. Here, we've already run it on those 4 samples, and you can see that each of these cost on the order of 40 to 50 cents each. So again, not a lot. So you can dig deeper in through this to see what were all the tasks and how long did it take. And for benchmarking and estimating how much would your job take, you can see where the bottlenecks are and where the efficiencies can be. So now that I've walked through this, what I want to talk about is how can we create this sample table right here? I'm going to switch to the analysis tab. And in here, we have created a combined data workspaces Jupyter notebook. If I just click on it, it gives me a preview of the code that's in it. But for me to run it, I need to go to my clone. So I'm going to go to the workspace that I cloned and start it up Jupyter environment. You can see it started because there's a green dot. And if I go to analysis and open up this notebook, in a second, as soon as it allows me, it's, I think my session logged out. So let me just re-log in here. Now I can click on the combined data workspaces. And if I click on open, it's going to copy that over into the Jupyter Notebook Cloud environment. And here now I have access to a Jupyter environment with all the relevant CPU, memory, and disk that I had provisioned. And now I can run through the code. As I mentioned, this is a package that's provided by the Bioconductor Consortia called AnVIL. And so if I run this code block here, it's going to load the packages into my environment. And what I'm going to want to do now is I'm going to want to pull in the data table from the HPRC consortia. And by default, for this example, I was pulling in 2. So 2 from HPRC, 2 from 1000 genomes. But what if instead of 2, I want to actually have 3? I can modify this code, run this block, and what's now going to show me in the output is that I've now pulled in the first 3 samples from HPRC. Same thing for 1000 genomes. Rather than just pulling in 2, I want to pull in 3 samples from the 1000 genomes. So now I have 3 from HPRC, 3 from 1000 genomes. And what I want to do is I want to update that data table. If you recall this data table here, I'm not going to click on it right now because it'll just stutter a little bit. It only has 4 samples. So what I want now to do is update it. Again, the Bioconductor AnVIL package has this AV table import. If I run that, it's now going to update it so it has 6 rows. So now if I switch back to data, it usually takes about a minute or so, but it's going to detect that there was a push through the API to update this table. And oftentimes what I see is that there is an indicator saying, hey, we are updating your table. One thing I realized that I actually didn't do is I didn't push this to the correct workspace. I pushed it to the original rather than the copy. So let me go back to the original workspace and log into here as well. Go to the data, and you see it says data import is in progress. So once this data import finishes, this will give me a dialog box saying it's ready, hit refresh, and this will go to 6. Again, the reason why I had this little hiccup here is because I pointed it to the original workspace, not my clone. Any of you running this won't be able to update the original workspace because you don't have that permission. And so you would just get an error saying you don't have access to update that set. So now you see the data has been imported successfully. I'm going to click refresh as it suggests. And now you can see that there are 6 samples. There's now 3 from HPRC, 3 from 1,000 genomes. So you can do this. You can use the AnVIL bioconductor package, or you could do any other way to create this table that you see fit. Now that this has been imported, I'll be into the other workspace, I'm going to show you how you kick off the workflow. So as I mentioned, this is the CCDG QC analysis pipeline. We've already imported it. And once you open up that workflow, what you're presented is the inputs tab. It allows you to configure this with various settings. There's many of these. You can learn more through the documentation, but I'm going to point out 3 of them. One is the input, what BAM file do you want to look at? We're saying that we want to look at the CRAM column. So if you recall in the data, we had 3. We had the sample, we had the CRAMs, and then we had the consortia they came from. So CRAM is what we want to look for. The second is what reference genome are we using? Here, the reference FASTA. You can see that we're using HG38. This is, again, what the pipeline is expecting. And then here, there's another setting called preemptible tries. This is one way that you can decrease the cost of your analysis by using preemptible environments, saying that it's okay if someone else needs the resource, I'm willing to pay less, that if it gets canceled, I'll just restart it. Again, AnVIL will take care of all that for you. Here we're saying try it 3 times before giving up. And you can, again, adjust this as you see fit, depending upon how you want to be paying for this, where you have to optimize if you need speed or if you just want to minimize costs. So all those settings, the defaults should be fine. We've configured it as such. What you need to do is select what samples you want to run on. So here I can click on select data. I can say that I want to do 1 from HPRC, 1 from 1,000 genomes. And I'm going to say, okay, those are the 2 that I want to run this analysis on. Now that I've selected the data, as you see 2 samples, I click on run analysis, and it's going to give me an option to give a note saying run QC analysis pipeline on 1 from HPRC and 1 from 1,000 genomes, right? And I'll say this is try running. And this is, again, just a note for me so that I understand why did I run this job. I'm going to click on launch. And now behind the scenes, it's going to start submitting. Just like on a cluster, you submit it to the cluster, it's in queue, it'll turn into being submitted, it'll turn into running, and then it'll turn into either succeeded or it failed. And if it's failed, you troubleshoot. If it succeeds, then you can go and look at the output, again, organized in the data tables. So at that point, I'm going to sort of stop right here because I want to just recap what I showed you today. What I showed you was, again, how you can organize data across multiple workspaces from different locations, including your own, using data tables, how to create it using the bioconductor AnVIL package, and how do you actually kick it off using a workflow, in this case, a QC analysis pipeline. I didn't show you how to find datasets using the dataset catalog or how to look for workflows in DocStore, but I'm more than happy to show you, if people have that interest. I'm going to reemphasize that all of these instructions are available in our AnVIL's demos repository, We're continually updating this with more demos. As Natalie had mentioned, we have more talking about Bioconductor and BioGalaxy. We'll be putting more of these here. You can work through them asynchronously at your own pace. You'll have links to the workspaces slides. If you're just in general interested in learning more, again, we have our AnVIL in 2 minutes shorts on YouTube. Highly recommend that to get a big high-level overview of what features you may be interested, and then finding out more. And as you have questions, ask us at our support forum, help.anvilproject.org. We're more than happy to answer any questions, point you in the directions that you have. If we're able to answer it, we will. If not, we'll try to connect you with people that can. With that, just like to briefly acknowledge, especially Ali and Ava, who helped pilot a lot of this last week for our board meeting, as well as Katie, who's been helping get a lot of this material available on our AnVIL Demos repository. So with that, I'm going to stop screen sharing and open it up for questions.
Thank you so much for that presentation, Fred.